8.2.2 wants us to outline the process of glycolysis. Now, you remember we talked about glycolysis. Glycolysis simply was, right, taking glucose in core, right? We talked about it, and changing it, that's supposed to be a 2, into 2 molecules of pyruvate. And when we do this, we get a net gain of 2 molecules of ATP. So that was glycolysis. But we need to know a little more. So when we talk about this, they would like us to talk about there's 3 processes that happen. There's a phosphorylation step, there's a lysis step, and there's an oxidation step. And also this ATP part gets a little bit confusing. All right, so let's dig in. So here is our 6-carbon molecule. Okay, it's glucose, but it's been like stretched out into a straight line, and they just call it hexose. So now this hexose molecule has 2 phosphates added onto it. So phosphorylation, if you want to think of this, is just phosphating it. It's not a verb, but if you want to think of adding phosphates onto something, we use this word phosphorylation. So what are we adding the phosphates to? We're adding them here, right? We've added phosphates to each side of our hexose sugar. Where did we get those phosphates from? Our ATP. Wait a minute, Mr. Fleming. I thought we got ATP out of this deal. Well, in this first step, we actually it costs us 2 molecules of ATP. So we've lost 2 molecules of ATP down here. Okay, so we've phosphorylated our hexose. So now we have a hexose phosphate, but there's 2 of them. So it's called a hexose biphosphate. I'm going to turn off the refrigerator real quick. The refrigerator was so noisy. Yeah, so we've created this thing, hexose biphosphate. You don't need to memorize these. However, in the long response on paper 2, you do get points if you can remember the names of those. Okay, so step 1, check. Step 2, we're going to lyse it. Remember, lysis means break. And we're going to cut this thing into 2 molecules of a 3 sugar compound, 3-tri-sugar-ose, triose, and there's a phosphate on each one of them. So it's a triose phosphate. Not terrible to remember. No, maybe. And then the last thing here is 2 things are happening for us to finally create our pyruvate molecule. Here, we're going to take 4 molecules of ADP, and we're going to get, in the end, 4 molecules of ATP. So now we're plus 4, and there, 4 minus 2 equals 2 for our HL math students. So that works out. We have 2 molecules of ATP in the end. What about this oxidation part, right? So that's our last step. Remember, oxidation is gain. And what do we gain? Electrons, but more importantly, these protons. And if you look down here, we've oxidated, right? We've oxidated the NADH, so now it has a hydrogen on it. That's it. That's how it goes. Except in the end, right? We have our pyruvate down here. A 3-carbon molecule. The phosphates are taken off. We've got 2 of them, and this will eventually, if it's aerobic, go into the mitochondria for that whole Krebs cycle thing. And if there's not enough oxygen, this pyruvate will be changed to lactate in animals, and if it's a yeast, it will be turned into carbon dioxide and ethanol.